Yeah, I'm very excited to share my study, and I'm very glad to see a lot of new faces that I haven't been seeing previously, and I'm impressed by there is a large community on fibrinolysis in Australia. So I will be talking about my recent studies in connecting the lipids and lipoproteins with the fibrinolysis system. So we all know that TPA binds to the fibrin clot, and where they convert plasminogen to the active enzyme plasmin, which degrades the fibrin clots. And TPA can be released from the endothelial cells upon acute injury, and PI1 can bind to TPA, form a complex, and therefore inhibit TPA function. So a few years ago, I described a study that showing the hepatocyte, the major cell in the livers, can make TPA and also secrete the hepatocyte-derived TPA into the circulation and also contribute to fibrinolysis. So specifically, we used a model to investigate the role of hepatocyte-derived TPA by using the AAV H1 vector to deliver the hairpin RNA against TPA mRNA in the hepatocyte. And you can show, you can see that within this model, the liver TPA mRNA reduced by 75% without any change in the endothelium TPA mRNA levels. And using this animal model, we found that the hepatocyte-derived TPA can contribute to the basal plasma concentration of TPA and also contribute to the TPA enzymatic activity and also fibrinolysis. So as TPA is a serum protease, we would assume that if there is more protein, it should correlate with the higher enzymatic activity. However, for TPA, it's not always the case, because if you look at the human plasma, TPA enzymatic activity actually negatively correlates with its own antigen levels. So how this could be? The TPA enzymatic activity negatively correlate with the PI1 antigen level, which makes sense because PI1 is inhibitor. But what's really confusing is that if you look at the TPA antigen level, it actually positively correlates with its own inhibitor PI1 antigen level. So we had the question that whether PI1 can actually not only inhibit TPA, but also induce a compensatory increase of TPA. So to really prove this concept, the first experiment we did was just simply inject recombinant PI1 into the lean mice where the PI1 circulating level is relatively low. And shortly after 1.5 hours, the TPA mRNA level in the livers actually goes up. And we also observed a similar result in the human primary hepatocyte in culture when we treat them with the recombinant PI1. We then use obese mouse as a model because in the obese mouse livers, there is a very high increase of PI1 by 12-fold increase when you compare the obese mouse liver to the lean mice. And the TPA mRNA level only goes up by 3-fold. And so the net liver enzymatic TPA activity is reduced in the mouse livers. So then we want to know, like, really whether the PI1 is the stimuli for the TPA expression level in the livers. And to answer that question, we use a hepatocyte PI1 specific knockout mice under the obese condition. And the question we want to know is if we remove most of the PI1 from the livers, and while the TPA enzymatic activity and fibrinolysis activity are increased, what happens to the TPA antigen levels? And so we actually observed that in the PI1 hepatocyte knockout mice, the plasma TPA antigen level is reduced while the TPA enzymatic activity are higher. So this really consistent with our hypothesis that the PI1 can actually serve as a stimuli for the TPA expression. Then we want to really think about the larger picture, and we for this study. So we all know that ischemic heart disease and stroke are the leading cause of death and accounts for a quarter of deaths globally. But if we look at the pathology of those two diseases, they actually share two common features, that they both can be initiated by the accumulation of lipid-rich plaque on the arterial wall, and they both can be triggered by the formation of blood clot. So the bigger question we are interested is, are the blood clot and lipids just coincidentally occurring in those two diseases, or there are mechanistic link connected the two process? And also, to specific to our study, what is the physiological role of having TPA expressed in the hepatocyte? So is the hepatocyte TPA only just provide a basal, as another basal TPA cellular source? So we did a literature review, and we found that in the clinical observational studies in the past few decades showed that the blood TPA enzymatic activity is actually negatively associated with the LDL cholesterol levels in patients with cardiovascular disease. So our question is whether TPA, in addition to its known function of breaking down the blood clot, can actually lower LDL cholesterol levels. Before I talk about our study, I want to briefly introduce the concept of lipoprotein assembly. So VLDL is the largest ApoB 100 containing lipoprotein, and is only assembled in the hepatocyte. The blue color are the ApoB protein that is holding up the structure of this lipid particle, and there is only one copy of ApoB per particle. Once VLDL is assembled in the hepatocyte, it will be released into the circulation, where it gets hydrolyzed by releasing the triglyceride to the peripheral cells and tissues. And then the VLDL particle will shrink to the smaller size, to the IDL, or eventually to the LDL. And all of the ApoB containing lipoproteins are considered as atherogenic. And in the past decades, there has been drugs developed in lowering the LDL cholesterol, including the statins. And statin can actually reduce LDL cholesterol levels to very optimal levels. However, with the optimal LDL cholesterol achieved in the populations, the cardiovascular risk is not really optimal. So therefore, we had there is considered that one of the approach to further reduce the cardiovascular residual risk is we have to lower all the ApoB containing lipoproteins, not only the LDL cholesterol. And if we really think about why hepatocyte needs TPA and what's the physiological role, so one of the idea is having TPA in the hepatocyte maybe give us an opportunity to think about its intracellular function on the VLDL assembly. So we first did a study in just simply silencing the hepatocyte TPA in an atherogenic mouse model. And from the cross-section of the mouse aortic root, you can see the atherosclerotic lesion is enlarged in the hepatocyte TPA silenced mice. And the total cholesterol levels are also increased in those hepatocyte TPA silenced mice. And the fractions having the higher cholesterol are actually on the ApoB containing lipoproteins, including VLDL, IDL, and LDL. All of those are atherogenic. So there are two major mechanisms determine how much VLDL can be secreted from the hepatocyte. One is how many particles are made, and the second is how well each particles are made. So to answer the how well each particle is made, we can measure the size of the VLDL particles released from the hepatocyte. So we isolate VLDL particles from the human primary hepatocyte culture medium, and we compare the size of those particles using the dynamic light scattering method. And after silencing TPA, the VLDL particle diameter increased. We can also visualize those particles that isolated from the mouse plasma and using the scanning EM. And you can see that after silencing hepatocyte TPA, the VLDL particle diameter is increased. So triglyceride is the most abundant lipid in the VLDL particle, and there is only one ApoB per particle. Therefore, we can use the triglyceride to ApoB ratio as a surrogate to indicate how much of lipid per particle are in the VLDL. So we found that after silencing TPA, the TG to triglyceride to ApoB ratio are higher. So there is all of those data suggesting that there are more lipid per ApoB particle after we silencing TPA in the hepatocyte. So what's the mechanism? And then I have to explain how the ApoB are get lipidated in the hepatocyte. It's mostly happening in the ER of the hepatocyte. And ApoB, when it's first made, it doesn't have lipid on it. And there's another protein called MTP, or the macrosomal triglyceride transfer protein. It's not only transferring triglyceride, it actually transfers all of the neutral lipids, including cholesterol and phospholipid and triglyceride. So the MTP shuffles all of those lipids onto the nascent ApoB and start making the VLDL particle in the ER of the hepatocyte. And we use a lipid transfer activity assay to monitor how much of the fluorescently labeled lipids can be transferred from the donor vesicle to the acceptor vesicle. And this lipid transfer activity can be reduced after we add the recombinant TPA to the reaction. So then the next experiment we did was isolate the ER fraction from the human primary hepatocyte and isolate the ER fractions by density, because if the ER has more lipids, it will be lighter. And comparing the ApoB 100 accumulation in those different density ER fractions, we can see that after silencing TPA, most of the ApoB are accumulated in the lighter fraction of the ER. So this is suggesting that after TPA is silenced in the hepatocyte, the ApoB gets more lipids. And we also want to make sure that the two proteins are interacting with each other and where they are located at in the hepatocyte. So based on the confocal imaging, we saw that not only the TPA and ApoB are co-localizing in the hepatocyte, but most of them are co-localizing with each other in the ER. So what's the mechanism? We know that the TPA has a lysine binding site on the Kringle2 domain. And ApoB actually has a lysine exposed region on its N-terminus where the ApoB can be lipidated by the MTP, the lipid transfer protein. So the hypothesis is that whether the TPA lysine binding site can directly interact with the lysine residues on ApoB N-terminus and then block the lipidation of ApoB. So the blue line is just the LDL and TPA interaction, and we can see they have a very strong binding. However, the red line is after we add the TPA Kringle2 domain-specific blocking antibody, and we can see that the interaction is blocked. So then what's the physiological relevance of having this TPA and ApoB interaction, especially if we put into the concept of hepatocytes? One of the major roles for the hepatocyte is handling the dietary lipids loading and then reassemble them into the VLDL and send it out to the rest of the body for the nutrients utilization. So, and we also want to know what is the role for the PAI-1. To mimic the lipid loading in the hepatocyte, we use oleate acid as a model. And when we load it to the hepatocyte, because oleate acid is the most abundant fatty acid in our diet, and it's also known to stimulate VLDL assembly. So after loading the oleate to the human primary hepatocyte, you can see the TPA's molecular weight shifted from the free TPA size to the larger size. And we believe that the larger size is the TPA PAI-1 complex. And this is also confirmed by the ELISA that showing after oleate treatment, the TPA PAI-1 complex are increased while the free TPA is reduced. Then we want to know whether this mechanism is really conserved in the humans, not only in the mice. So we collaborate with Dr. Amy Shapiro and her colleagues at the Indiana Bleeding Institute, and where they treat the patients with homozygous PAI-1 deficiency. And you can see in those patients, their LDL cholesterol is lower. But more interestingly, that when we isolate the VLDL particle, they have higher TPA associated with those particles. And if we do the correlation for the VLDL particle size, it actually have a negative correlation for the larger VLDL particle will have less TPA associated with them. So this is consistent with our hypothesis that when you have TPA, the TPA can, in the hepatocyte, the TPA can reduce the ApoB lipidation in the basal conditions. But once the lipid is loaded into the hepatocyte, the PAI-1 can sequester TPA by forming a complex and release the N-terminus of ApoB that is accessible for the MTP to shuffling lipids onto the nascent ApoB to start forming the VLDL particles. This study was led by Dr. Wen Dai, who is an outstanding postdoc fellow and now research scientist in our lab. And he moved on after finishing the VLDL study. Then he's answering another question that there are actually two major types of ApoB containing lipoprotein particles. One is the VLDL and its subsequent LDL particles. Another major one is in the chylomicron, which is only made in the intestinal epithelial cells. So the question is, what's the role of TPA in the chylomicron assembly this time? So he made the intestinal epithelial TPA-specific knockout mice, and we challenged the mice with the olive oil gavage. And you can see that under the basal condition, without this olive oil gavage, there is no plasma non-esterified fatty acid level difference. However, after the olive oil gavage, the intestinal epithelial TPA knockout mice has a sharp increase for the non-esterified fatty acid levels. And I'm going to close up my talk with the last piece of data in collaboration with Dr. Dan Lawrence and Joe Su in his lab, that we used the PAI-1 inhibitor from Dan's lab and see whether it will have opposite effects from the TPA knockout mice. And consistently, after treating the mice with the PAI-1 inhibitor, and we also gave them the olive oil gavage, and this time the inhibitor-treated mice have the the reduced fatty acid in the plasma after the olive oil gavage. And however, they actually have increased feces fatty acid levels. So this together with the intestinal TPA knockout mouse result that we believe TPA can reduce the chylomicron assembly in the intestinal epithelial cells, and PI1 have the opposite role. But there are a lot that we need to further validate this conclusion. In summary, we found that hepatocyte can make TPA and it contribute to nearly half of the plasma TPA basal concentration. And in the obesity condition, liver have an imbalanced TPA and PI1 production, and leading to reduced TPA activity and fibrinolysis in the obesity conditions. Within the hepatocyte, we found TPA can directly inhibit the VLDL assembly in the ER, and PI1 inhibition can reduce the postprandial fatty acid levels in the plasma, while the intestinal TPA has the opposite role. So I only talk about the TPA studies, but we actually have several other studies together to answer the question for the connections between blood clotting and lipoproteins. So we actually have several different projects, including what is the role of bile acid in this mechanism, and also for the coagulation using the hemophilia as a model, and also platelet. So I will close and acknowledge everyone contributed to this study. Thank you.